All right, we're going to go ahead and begin to get started. I see that Dr. Moore is here with us as well, and Charisse is here too. So awesome. So our beginning folks are here, and we want to sort of stay true to time as much as we can because y'all, I just heard the titles, and I'm excited. And so I know to hear more from our students. We are excited to welcome everyone. This is our second year of doing this McNair Symposium with the support of the U.S. National Science Foundation, as well as the Department of Education. And so in partnership with the UMBC McNair Scholars Program. And so we welcome you all. We're excited to have you. Thank you. This year, we also are excited that we've expanded this opportunity. So you'll sort of see folks in the middle screen. It's called OWL. So those, if you are in the ballroom, just wave so folks can see you. I see y'all in the ballroom. So we see folks there. And so we are very excited to have our students who are actually attending the conference here in Baltimore. They'll be watching and engaging. We also offered folks to join us online. So we have folks online here with us. And we're going to go ahead and begin to get started. We do this because we want to express the profound excitement that we have that our scholars are actually thriving in research, and to hear, to have our national leaders hear from them, and to also for you all in the sense of reciprocity where they'll be able to hear from you as well. And so as we get started, I'm excited that we have some of our national leaders with us, and we're going to ask them to provide greetings as we begin this symposium. And we're going to start with Dr. Moore, followed by Charisse Carney-Nunes, and then followed by Dr. Norman.
Good morning, everyone. I am so happy to be a part of this symposium. I look forward to hearing more about the profound research that you all are doing. I know that the research that you're doing will change lives, will make an impact in our world. But first, I just would like to bring greetings on behalf of the U.S. Department of Education. Our Senior Director, Gaby Watts, is committed to the TRIO programs and all of the programs that we serve under our portfolio because we know that TRIO works. We're celebrating 60 years of changing lives and providing hope and opportunity, and therefore changing generations. And for that, I thank you all for all that you're doing. And I would just like to encourage you to continue to make the needed sacrifices, stay committed and dedicated to the work that you're doing. And lastly, I would like to just thank you, Michael, for all of your hard work with what you're doing with our McNair scholars and for all the McNair project directors that are there. Please continue the hard work that you're doing. It is making a difference. Thank you again for this opportunity. Excuse me. Thank you again for the opportunity.
Go ahead. Okay. Hi, my name is Charisse Carney-Nunes. Good morning, everyone. I am the Deputy Division Director for the Division of Graduate Education within the Directorate for STEM Education at the National Science Foundation. I bring you greetings on behalf of NSF. I'm the Deputy Division Director. Our Division Director is Jacqueline Jacqueline Huntoon, and then our Assistant Director is Dr. James Moore for the Directorate of STEM Education. In my brief welcome this morning, I wanted to remind you today to practice the 3 L's: listen, learn, and lead. And I wanted to do that because of just the incredible number. This is my second year doing this. I was so excited when Dr. Norman invited me to do it again. So first, in terms of listening, NSF is really excited to have this opportunity to learn about your research and to share this opportunity with our colleagues at the Department of Education. Listen to everything that you hear today from your peers, from us. Just listen to all voices. Next, learn. You will hear us talk about many opportunities that are available at NSF for every step in your academic careers. So don't just listen, but take the next step and learn about them. A couple of those that are most relevant to those of you participating in this event are the NSF Education and Training Application, known as ETAP, and the Graduate Research Fellowship Program, GRFP. I wanted to pause and tell you a quick story to just connect with you, to let you know that I am the Deputy Division Director of Division of Graduate Education, but I'm also a mom. I'm a mom of students just like you. And my student is a sophomore STEM major, majoring in engineering. And I'm just going to tell you that he and his friends love ETAP. When I told them about ETAP, they were like, what? Because ETAP is still kind of new, but just make sure that you just take the extra step and learn about everything that it has to offer. I know that my son and his friends got research experience opportunities just this past summer through what they were able to find and aggregate easily on ETAP. And it's just a game changer in terms of just access, finding out more easily finding access to opportunities that we have through NSF. So they love it. And then, of course, the GRFP, you'll hear so much about that, but just take the extra steps. My best advice pertaining to learning about the GRFP is for you to read everything. And I say that with emphasis, everything. Read, follow all of the rules. And that's really the best advice I can give about GRFP. So you will hear more about both of those and much more later in the symposium today. And then finally, just lead. Take everything that you listen and that you learn about back into your everyday lives, internalize it, put it into action. Remember, you are the leader of your own life, your career. Share it with your friends. You can be a peer leader. You see my Zoom background. We are stronger together than we are separately. Thank you for spending the next few hours with us. And we hope that this will help increase your interest and participation in NSF programs and activities now and in the future. So thank you.
I just like to add just a little bit. I am Akriti Norman, very excited to be able to speak to you all today. Not just because the GRP program is a group of fans actually of McNair Scholars, because we know about how hard you work and we know about your contribution to STEM, but also because these type of events are mutually beneficial. When you work behind the scenes in fellowship programs like I do, you don't often get to see those results and those students and the student research. So it's mutually beneficial. It keeps me motivated and gives us all energy on the NSF side. So I'm looking forward to not only talking to you more about NSF, but hearing about your research.
Awesome. Once again, we thank you all for this partnership and support. I want to say that, and I lift this in this space because every time I come, students, but then I think I learned stuff from NSF. Like there were things that I learned last year, such as the new portal and everything that you all have, that I was like, oh my gosh, didn't know this existed. And so again, I myself, as a director, am very excited to be here again to hear on both ends. And as we get started, we'll be starting momentarily, but I wanted to put into the chat. Let me do that now. Everyone in meeting. Oops, wrong thing. Here it is. Yes. Okay. This is just our website that if you scroll down, you'll sort of see the order of the day. And so we're trying to keep in with the time. We had chosen and then one didn't respond right away, but then responded, and I was like, well, okay, now we got 9. So we started messing with I said, okay, I put them on a wait list, but then I said, no, this is just so great. I just want to hear all of y'all, and I want to make sure that NSF hears, NSF and the Department of Ed hears all of you. So we maneuvered the schedule a little so that we can get all 9 students in. And so with that, our goal is to try to stay on time in the process. And I've already talked with our presenters about this. And so I know that they're going to be phenomenal, and we're excited for that. We also do have, I think, and I need to check with my team, and if anybody on my team knows for sure, we may have a way for you to provide feedback to the presenters. But either way, the presenters, you all will have copies of this recording. We will make the necessary edits and then be able to send it over so that you all can actually see yourselves and be able to do some self-reflection on your presentation, as well as the presentation from NSF. We will be able to share minus the Q&A portion of their session. But we're grateful that we're able to sort of, again, give all information to everyone. Feel free to be on and off camera as you feel desired, but we're going to really center our time in hearing from our speakers. And so we want to get them ready to go. And as my growing up in the NAACP in Baltimore, there was anytime we were about to do some good work and some good trouble, as Dr. or Representative Lewis would say, when we get into good trouble, we would say, fired up, ready to go. So I want everybody you don't have to come off of mute, but I want you just to hear you say, fired up, ready to go. We're fired up and we're ready to go. And so we're excited to hear your presentations, because you're all about to present things that in the end will change the world. And so we're going to start off our time here with Eve Butterworth. So Eve, you can go on, share your screen, and we will begin.
Thank you. Hello, everyone. My name is Eve Butterworth. I'm a senior at Wellesley College studying geoscience with an emphasis on environmental geochemistry. And this is my preliminary research for my senior honors thesis in collaboration with the National Oceanic and Atmospheric Administration, entitled Life in Plastic is Not Fantastic, an analysis of plastics and sediments in the Massachusetts Mystic River estuary. So my area of interest is an estuary north of Boston, Massachusetts. An estuary is a river connected to the ocean, making it experience tidal conditions. Urban estuaries in particular, like this one, are interesting because they can bring contaminants from the ocean, city, and the harbors through vectors such as flooding and runoff. My mission in this project on a broader scale is to understand what contaminants are in this estuary and where they may congregate. A better understanding of contaminants within this area can lead to better communication, better preparation, and better working together within the community, making for ultimately a more just community. So why did I choose the Mystic River estuary in particular? This is specifically an estuary in proximity to what Massachusetts defines as environmental justice communities. These are communities that meet at least one of the following criteria. At least 40% of the residents are people of color. The annual median income of these residents is less than 65% of the statewide median income, and or 25% or more of adults in homes here identify as speaking English less than very well, which is defined as English isolation. People in environmental justice communities have less access to safe natural resources, such as safe parks for their children to play in, clean air quality. They are more at risk for pulmonary illnesses due to air pollution, have more contaminated water, and also are generally more unsafe and bear an unfair burden due to environmental contamination. This estuary area in particular currently hosts shipping, manufacturing, and the storage of manufactured goods along the estuary, transportation over the estuary, which may result in runoff, and flooding potential, specifically due to climate change. There's also a history of naval use within this estuary, including harbors, massive citywide fires, manufacturing of coke as in coal byproducts, and metal smelting, which resulted in a Superfund site, or a site so contaminated that it required federal remediation. So the Mystic River estuary has several parts, but my location is this small area. The estuary's name, Mystic, comes from the indigenous name for the area, Misituk, which means tidal river or estuary. The estuary is somewhat tragically cut off anthropogenically through the Amelia Earhart Dam. As you can see, there's a slight color difference between the estuary side and the non-tidal side, as water on the estuary side is frequently replaced. My sampling at sites included a storm drain that ran off from a parking lot, a tidal mud flat, Phragmites reed patches, which is a common reed along the banks of many rivers, and sediment taken from above and below the dam. My research goals in this project were to identify common congregation points for plastics and contaminants within this estuary, to analyze and identify plastics both visually and chemically. This was done with NOAA, and to analyze sediments for possible contamination, both contemporary and legacy, specifically metals like lead and zinc. Starting off with my plastics, I collected sediments that we suspected contained plastics in 2 trips at low tide. I then separated the plastics from the sediments by hand under a microscope. And then I cataloged and photographed these plastics, mostly under a microscope as well, and prepared them for chemical analysis. Starting off with a visual analysis, I compared plastics from above and below the dam. Visually, I noticed that plastics above the dam had higher densities. They tended to be industrial materials and things like bottle caps and full bottles. Below the dam, we had much more light and less dense materials, things like fishing lines and styrofoam. We also had a higher congregation and concentration of microplastics, or plastics that are smaller than 0.5 centimeter. What ultimately ended up being a massive focus of my project were Phragmites reed patches. Phragmites are an invasive reed across the United States, choking out other plants on the banks of many of the rivers across the US. They're also a congregation point or trap for many types of plastics of all sizes. As you can see on the left, they can trap microplastics in the form of fibers, which are actually the most microplastic, all the way up to very large macroplastics like bottles and things like cellophane, wrappers, styrofoam, and everything in between. What became particularly interesting to me is the ability for plastics to attract and attach heavy metals onto them through a process called adsorption. These heavy metals can then piggyback off of plastics onto into traps like Phragmites patches where they may reside. This research is still pretty new and became an exciting new idea for a transport vector of contaminants like lead into a new sink or trap like Phragmites. So my analytical methods for identifying plastic I used pyrolysis gas chromatography mass spectrometry, or pyro-GCMS. The resulting spectrum looks like this, and it divides the constituent molecules of these plastics. This is polyethylene in its pellet form, which is often how polyethylene is transported before it is manufactured into its final product. My breakdown of different types of plastic found that the most common plastic in all of my locations was polyethylene, followed by polypropylene and polystyrene, which is a main ingredient, you could say, in styrofoam. The interesting thing about polyethylene and polypropylene is that they have the highest affinity to attract and absorb both lead and zinc onto them. And lead and zinc are two metals that I particularly am interested in studying, as they are both common and a common contaminant within this area. So moving on to those heavy metals, I used X-ray fluorescence to determine the element concentration in parts per million of various metals within this system, particularly focusing on comparing Phragmites populated areas with sediments nearby that were not Phragmites populated. Specifically, I was comparing above and below the dam. We found that the Phragmites areas had higher concentrations of most metals, specifically lead and zinc, versus their non-Phragmites populated comparative sediments. This does support the idea that there might be some connection between the Phragmites, the plastics, and the metals, particularly that it points to the potential for a transport vector. In conclusion, we found that Phragmites reed patches were a major congregation point for plastics and may also be a transport mechanism for those plastics. We also found that plastics showed some visual differences above and below the dam, but it should be noted that my results were limited due to some technical issues with my analytical equipment, and also bias because I am picking out those plastic samples by hand under a microscope. For heavy metals, we found higher concentrations in the Phragmites patches, which does support the hypothesis that Phragmites may be a trap for more than just plastics, and supports a connection between these three areas: Phragmites, plastics, and metals. This is going to be the main topic of my senior honors thesis. Moving on to the most important thing that ties us all together, I think. This is a very preliminary study, but we intend to utilize our skills and resources as a privileged institution to provide information and tools that can be used by communities to adequately prepare, protect, and advocate for themselves. As always, the priorities and concerns of community stakeholders are of the utmost importance, and communication between scientists and communities is not only key, but it is an obligation for scientists. I would like to acknowledge the Noah Holling Scholarship and the Wellesley College Geoscience Department for funding my research, as well as the McNair Program for allowing me to be here. A special shout out to the NSF and the Department of Education for specifically inviting me to be part of this symposium, and Jocelyn and Teo, the directors of my NSF program at Wellesley College. This project is specifically dedicated to the Indigenous people of the Boston area, where I research, collect samples, and study the Massachusetts, Wampanoag, and Nipmuc nations, and to all people living under colonialism and occupation. My CV and contact information is here if you'd like to scan this QR code. Thank you so much. Let me know if you have any questions.
Awesome, 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 awesome. Thank you, Eve, and really connecting it to your, it seems like your life work there. So really appreciate that, and you presenting. We want to open up to questions, so we got a few moments for questions. Any questions?
I can ask one question. Can you talk a little bit about your interest in absorption of lead and zinc?
Why I'm interested, or?
Yes.
Well, lead and zinc are very toxic in high concentrations, particularly to children. And there is a history of industrial activity and contamination in this area. So adsorption of lead and zinc onto plastics could be a transport mechanism moving this lead and zinc that we know is here into areas we might not know about. And then with flooding, that could move again in areas, again, we have no idea about, which means that it would be good to understand both how adsorption works within the system, because it is both pH and temperature dependent, and also overall where it is going.
Thank you.
Any other questions? We have someone in the viewing room. Yes. Can we hear them in there?
Hi, can you hear me?
Yes.
Okay. Hi, my name is Aliyah Bevins. I'm a senior biology major with a chemistry minor at Virginia Union University. And my research actually focuses on air quality monitoring and marginalized communities and like HBCUs and surrounding communities and stuff like that. And I was wondering if Eve thought there could be future directions in implementing like monitoring in like the water to give them a better understanding of it and stuff like that.
Absolutely. I mean, I think that monitoring should be done everywhere, and that we could always benefit from that data. Particularly, there's blood lead level testing that has been done here, because we're concerned about, lead in blood. But I think water quality and soil, particularly soil quality testing, would be massively beneficial.
Thank you. And I'm so impressed by your research and everything. You did so good.
Thank you so much for being here.
You're welcome.
Round of applause. Round of applause. Thank you, Eve.
Thank you so much, everyone.
All right. We will now move on to Dayna. Buitron. Go right ahead and share.
All right, you guys can see it and hear me?
Yes, you're good to go.
Great. Hello, my name is Dayna Buitron, and I'm a senior at the University of Idaho, majoring in microbiology with minors in biochemistry and philosophy. I have been conducting research in Paul Worley's lab, and I'm happy to present my findings in the investigation of the killer toxin binding interface to the plasma membrane receptor. Let's talk a little bit about yeast really quickly. Yeast are actually found everywhere. It's in the rising agent found in bread, such as this frog-shaped bread I made, and in cinnamon rolls. There are also single-celled microbes that have been domesticated for fermentation processes. Cacao beans have to be fermented with yeast to produce chocolate, same as your coffee beans for your everyday coffee. They are also a model organism for basic cell processes. Here's a GIF of yeast budding to get you a closer look at them. Yeast actually produce killer toxins that are involved in niche competition with each other. So here we have a killer assay. We have killer yeast pinned in the center. This killer yeast produces a killer toxin that interacts with the lawn yeast in the background, killing it. It creates this clear zone marked by cell death, is referred to as a zone of inhibition. These killer toxins can interact and kill other yeast, so that means hopefully we can have them target other yeast pathogens and create antifungals. It's important because fungal infections are on the rise. About every year, we see 150 million severe infections and 1.7 million deaths. This is an increasing number as multi-drug resistance is increasing. And we have, hopefully, killer toxins to help with that. Killer toxin K1 can kill Candida glabrata. Candida glabrata is the second most common cause of vaginal yeast infections. And Candida in general is the second most prevalent yeast pathogen in humans. And if we can get K1 killer toxin to target Candida glabrata and hopefully other Candida yeasts, then we can help further this antifungals to bring more onto the medical market. So let's talk a little bit more about K1 killer toxin. K1 is an extracellular toxin, and it enters the yeast cell right outside the plasma membrane. It interacts with the membrane binding receptor Cre1 to create a K1 pore. This K1 pore then has an efflux of potassium ions. This cation selective channel creates disruption of cell homeostasis, and this disruption is what kills the cell, and it's how we have that phenotype of sensitivity to K1. Understanding the specific interaction between K1 and Cre1 will help us understand how we can target K1 into other fungal pathogens. We understand Cre1 to be the membrane receptor because we can look at Cre1 delta. This is the lack of the Cre1 gene. This means that we are looking at a yeast strain or a system that lacks this protein. If it lacks Cre1, it cannot create this pore and thus no efflux of potassium ions. This means the cell survives, there is no zone of inhibition. Now, to put this into the lab scene, I have to understand how to control it. It's been a little difficult within my lab, but I finally figure out how I can control Cre1 expression to test how does Cre1 and K1 function together. So let's start with a little plasmid. It's a circular piece of DNA. I've inserted the Cre1 gene into it to create Cre1 expressing on a plasmid. So now I have Cre1 in any way I want on a circular piece of DNA. Then I insert it into a Cre1 delta yeast. So again, this is a yeast strain that lacks Cre1. This yeast strain is resistant to K1, and I am inserting Cre1 back in to kind of recomplement that phenotype that we see in wild type. And theoretically, we will have the zone of inhibition come out again. And we do. So now I can put my own Cre1 into a Cre null strain to test how Cre1 can really function with K1. And that's what I've been working on. So let's look at a schematic diagram of the protein really quickly. So the protein is characterized by 4 different sections, as you can see here. But we're really interested in the C terminus. Previous literature describes that K1 interacts with the C terminus at this specific section to generate this sensitive phenotype. However, the literature was not very rigorous in their findings, and I believe that there is a smaller domain of interaction. And thus this brings me to my main question. What is the minimal binding site for K1 intoxication? We need strict specificity in understanding this for drug therapeutics. So that's what I've been working on, and I have discovered a smaller region of interaction. Here, let's look at the carboxyl terminus again. This is our full wild type protein, so this is untouched, and it does have that sensitive phenotype. I've made the following deletions here. This was made with molecular biology and other methods that, if you would like to talk about, we can talk about afterwards. But we have 3 different deletions here. They are made right within the C terminus where it was predicted to bind, thanks to mathematical modeling. This first deletion right here, this proved that this section was necessary for K1 sensitivity, because when we remove that section, we see resistance to K1. For the second deletion, similar situation. This section is also necessary for K1 sensitivity. So therefore, we've now understood, at the very least, these 2 deletions are necessary for K1 and Cre1 interaction. This last deletion, however, is sensitive. Just a small halo around it, but enough to say that we definitely need these back 2 deletions, and this last one, not so much. So I've myself have brought out this 100-some amino acid theorized binding domain down to around just 40. Now, this is what I've done so far, and I'm working on improving this even more. So I'm closer to understanding this K1-Cre1 interaction, but I'm working with Dr. Marty Yitzerberg at the University of Idaho to further this work. He also looks at the K1-Cre1 interaction using mathematical modeling, and we've come up with predicted amino acids involved in this K1-Cre1 binding. So if you look here, this carboxyl terminus, I've now brought the theorized binding domain down to just this section. Me and Dr. Yitzerberg, we've found specific locations within this new small domain that can be changed to help understand what specific parts of this area are what's causing the interaction to occur and thus sensitivity. Now, this will help further, again, how K1-Cre1 functions for that possible therapeutic. Now, I'd like to acknowledge the NSF and McNair for allowing me to present at the symposium. My advisors, Dr. Paul Rowley and Dr. Marty Yitzerberg now at the University of Idaho. Idaho INBRE for supporting me during the summer for financially to support my research. The NSF Career Grant that my professors' work directly funds right now, and then the College Science Hill Fellowship and the Institute for Health in the Human Ecosystem at the University of Idaho that I have won to help support this funding as well. If you are interested in our lab, there's a QR code that you can scan, and we are accepting graduate students if anyone is interested. Thank you.
Awesome. Awesome job. And I appreciate, look, shout out to you for promoting your lab and getting more wonderful McNair scholars to join you even as grad students. So thanks for doing that. Shout out to your community, and especially since it seems that they have been instrumental in supporting you. So you want to, get folks there that can be supported as well. Questions that folks have. Any questions? Or questions or comments? Either one is fine.
I also like the shout out to your lab. Can you tell us a little bit more about why you enjoy being a part of that team?
There's a lot of factors that play into it. Other than just the research, I really enjoy looking at and understanding how these proteins function. I really enjoy the lab atmosphere. It feels very nice to go into lab and know people that I can talk to and I can just have fun around. It was my very first social group in college, so they were very fun and we were all very research oriented. So it was very nice to have a community that was also doing the same thing.
Thank you.
I would like to know more about how this research has impacted you in your own journey. So why this research path for you?
I originally reached out to Dr. Rowley to join his lab as a pre-med student as part of the research aspect of the medical application, the medical school application. And his lab is primarily undergrad ran. And for this, he has us write up whole research proposals to begin our work. So I was really in the deep of research of literature and reading about it. And once I started doing that and writing up my proposal and I was accepted into the lab doing wet lab work, I realized that I actually much more enjoyed research than the prospect of medical school. And I've been involved in some medical school positions, but I just found this one to be much more alluring. And as I've continued it and taken more classes, I find that I really do enjoy research and learning about these cell processes. So I would like to continue that into graduate school now. And it's thanks to this lab.
Awesome, thank you. Any questions or comments from those in the ballroom? I want to make sure I didn't miss anybody. All right. All right. Well, thank you so much. Let's give a round of applause.
Thank you.
Dayna, we really appreciate you sharing today, and we're going to keep moving. It puts us a little bit ahead, but that is great. Anthony Merchant.
You can see my screen, right?
Yes.
Okay.
It's not on full screen yet, though.
Yeah, okay, that's perfect.
There you go. Perfect.
Okay, my name is Anthony Merchant. I am a senior at the University of Oklahoma, and my major is aerospace engineering. And the project I'll be discussing today is on the radiometric calibration of the MASS-KMZ cameras on the Perseverance rover, and specifically on using the Martian sky as a calibration source. Mars 2020 and the Perseverance rover. Perseverance was launched by NASA with the aims of exploring Jezero crater to the left for signs of past biological life, collecting rock samples for future return to Earth, and to demonstrate new technologies for future human exploration. To the right is a picture of Perseverance and its companion Ingenuity. Key to the Mars 2020 mission is the MASS-KMZ camera system. MASS-KMZ is a pair of cameras that contains identical optical assemblies and has filters that range from the visible to the infrared spectrum. MASS-KMZ is actually the first zoomable camera sent to Mars, and it takes high-resolution multispectral images of the Martian terrain, which is essential for things such as rover navigation and conducting remote geological and compositional studies of the red planet. For MASS-KMZ, though, to deliver precise data, in-flight calibration is necessary. The environment that was originally used to calibrate the cameras is entirely different from the environment up in Mars, which means that new calibration is necessary. The picture to the left actually shows MASS-KMZ being calibrated in 2022 at Cornell University using lithosphere. And the picture to the right shows the environment that the cameras are presently in. Key to this calibration is a method called sky flat field correction. Sky flat field corrections play an essential role in this calibration, specifically sky flats. What is a sky flat? Sky flats are basically images of the Martian sky taken under uniform lighting condition. The picture to the right is actually one of multiple sky flats taken by the camera on Sol 1216. Sky flats are important for a variety of reasons, but the two main reasons is that one, it accounts for the minor differences that might occur in the physical alignment of the camera lenses because of the temperature and pressure differences between its original environment on Earth and its new environment on Mars. But it also accounts for things such as pixel-to-pixel sensitivity differences that occurs and dust and debris that might accumulate on the camera lenses. The methodology for my project was to first generate sky flat field correction. This included generating sky flats for Sol 35 to Sol 1216. A Sol is basically a solar day on Mars, or like a day on Mars, and it's basically used to measure the mission length. Sol 1 was the first day of the mission, and Sol 1216 was about 40 days ago. After generating these sky flats, we reviewed the sky flats, including validating the pre-existing sky flats generated previously for the first 700 sols of the mission, and reviewing the new sky flats generated for the last 500 sols. After this review was done, we updated the calibration pipeline, which included removing any erroneous sky flats from the recalibration pipeline and adding any new additional sky flats to update the pipeline at ASU and Barbados. When MASS-KMZ generates sky flats, it always starts off by identifying a central test area in the sky. This test area is always 180 degrees azimuth from the sun, which basically means it's facing away from the sun at a 30-degree elevation. After identifying the central test area, it proceeds to take a mosaic of highly overlapping images around this central test area. These highly overlapping images ensures that any focal plane effect is averaged out, and it's also used to derive something called a sky radiance map, which will be used later. After the sky radiance map is derived, using its 26mm camera, it takes a central frame. This central frame serves as the baseline frame as it goes from its widest to its narrowest zoom. Each image taken in each zoom position is divided out with the earlier derived sky radiance map to generate what we call a sky flat field correction. This is an example of a sky radiance map. It was called 1216, and these actually maps tend to be very pretty, and it tells us if the process was done correctly. This is something like you expect to see, so this is a good sky radiance map I decided to include. After generating sky flats for Sol 3016, we did a sky flat comparison. I specifically included two different zooms on the same on the right camera. To the left, we have the ground calibrated sky flat. These were calibrated and created at Cornell University. The reason why you see the high gradient around the sphere specifically points a light at the middle of the camera, which is why you see it like that. The next sky flat is a Sol 35 sky flat. Sol 35 is actually the first time the cameras were turned on. So it's called the baseline sky flat, and it basically measures the initial reaction of the cameras to the Martian environment. Any preceding sky flat are called delta sky flats because it now measures the effects of the Martian environment on the cameras as the mission continues. So I included Sol 484 and Sol 604. These are about 200 days apart. And as you can see highlighted, you see an increase in contrast, and that's kind of what you expect to see because as the mission goes by, the cameras accumulate dust and debris and stuff like that. This is another GIF file. As you can look at the right, you keep seeing higher contrast as the mission goes by. This is expected and this is normal and this is what you see. And this is why this process is done to correct for any debris that might get on the cameras. This process isn't perfect though, and specifically the sky flat generation process. And we have things called erroneous delta sky flats. This can occur for a variety of reasons, but usually occurs when clouds end up actually getting in the process. As stated earlier, sky flats have to be uniform images. So when clouds or like the sun enters at a weird angle, you end up getting weird images. And I included a raw image file and a calibrated file, and you can see what happens when this happens. Figure 5 specifically is an erroneous sky flat. This was taken on Sol 746, which happened to be a very bad set because clouds were actually all over the data set. After generating these delta sky flats, we reviewed and corrected them. As stated earlier, the previously generated sky flats for this first 700 sols of the mission were verified to be correct. But we also created new sky flats for Sol 700 to Sol 1216 to account for the new changes identified in the delta sky flats. The new sky flats can only have a maximum 2% gradient, and anything with a more than a 2% gradient was discarded, as you can see in Sol 1039. Sol 1000 and Sol 1115 show two perfect sky flats. You kind of expect to see that, but on Sol 1039, you see a yellow hue on the top left of the corner. That's not normal, and that's when you discard the sky flat. After generating these sky flats, we did a sky flat review. We created tables to account for each sky flat for each filter and each zoom position. I decided to include two different tables. I included a sky flat review table for Sol 799 and a sky flat review table for Sol 945. 799 shows what you expect to see. This process tends to actually be very perfect and streamlined, and you expect to see good sky flats. So this was a good table. But I include sky flat review table for Sol 945 because this was actually a really, really bad data set. The whole data set had to be discarded, and this was because the sun entered the camera lenses at a very weird angle. So in summary, the recalibration of the MassCamZ cameras started off by generating the new sky flats for Sol 700 to Sol 1216, with the previous 700 sols being verified to be correct. Sky flats for Sol 746 and 945 were entirely removed from the recalibration pipeline, while additional sky flats from Sol 849, Sol 896, and 1039 were also excluded. The remaining sky flats were basically used to update the pipeline at ASU in Barbados, which has updated and recalibrated the cameras on the Perseverance rover, which is presently on Mars. Why is this important? To bring all this together, a lot of you guys might be asking why this is important. In the famous words of Neil Armstrong, that is one small step, but one giant leap for mankind. Every calibrated image serves as a small step, but collectively, it is a giant leap for making a lot of discoveries and also unraveling the mysteries of our neighboring planet. I included 3 important images that Perseverance has taken during its progression of its mission. The left one is an image of the first time Perseverance drilled into the ground and collected a rock sample. This is a huge part of the mission, and it's supposed to be someday recovered and sent back to Earth. Second image I included is actually one of Jezero Crater's delta. A huge part of the Mars 2020 mission was to finally confirm that there used to be liquid water, and specifically liquid oceans on Mars. Jezero Crater was suspected to have once harbored an ocean, and we were able to confirm through Perseverance mass TMZ cameras and its remote geological and compositional studies that an ocean once existed on Jezero Crater. And the picture to the right is actually a recent picture. This was taken about a month ago, and it's a picture of a rock with leopard spots. To a normal person, including me, I actually didn't really know the meaning of this picture, but this might be one of the most significant discoveries of the 21st century. On Earth, a rock with a leopard spot is typically a sign of past bacterial life. And obviously this isn't confirmed yet, but this was a huge monumental discovery for NASA as of now. And Perseverance, taking this picture, decided to also drill into it and collect a rock sample. We won't know and be able to confirm NASA's susceptibility of what it expects until the rock samples are collected and sent back to Earth. But it's part of the mission, and it shows why it's important to ensure that Perseverance, and specifically mass TMZ, has precise and also perfect pictures. And that is the end of my project. I'd like to acknowledge my funding, which was Cornell University's Futures Leaders in Mechanical and Aerospace Engineering program. I'd also like to give a special thanks to Professor Alexander Hayes and the Compassi research groups for their support and guidance throughout this process. And a special thanks to Dr. Sophia Bolandillis and the OU McNair program for their support and guidance. And also a thanks to the Mars 2020 project and the Mass TMZ team for the opportunity to be a part of this project. That is it. Does anyone have any questions?
Phenomenal. Wow. This is, yeah, this is great. And even the pictures you showed at the end, I didn't realize that the bacteria one, that was cool. So craziness. Okay. Questions or comments?
I could ask a quick question.
Yes, go for it.
Anthony, what do you think about, what are your thoughts about ways to improve the efficiency of the SkyFlat process?
That's actually something that's been discussed like heavily. So the generating SkyFlats took a really, really long time. I think generated over 5,000 SkyFlats. And to process a solar day, it'd take me about 24 hours, if not more, depending how big the data set was. And we got into the problem that like sometimes I'd have to have the lab computer running things and then my computer, and it became very hectic. So we've done a lot of methods, iterative methods, where we'd edit the MATLAB code, because a lot of this is done through MATLAB and Fortran, specifically coding. And we've tried to streamline the process and the coding process, but it's like you can't remove you can only remove so much in that like without affecting the process. And we've kind of reached that point where like if we remove something, it might mess up the process or might not make the pictures as precise as we need them to be. So I'd say like we could streamline it, but I feel like it's been streamlined, I guess, as much as we can streamline as of right now without affecting the cameras necessarily.
Thank you. That's a great observation.
Any other questions?
We have a question in the viewing room.
All right, go for it.
Hey, Anthony. So how has this project had an impact on your future, and what kind of profession you're going to be interested in going through grad school and then once you come out?
Actually, this project has impacted me a lot. So for the longest time, I was actually unsure of what I wanted to do with my degree, specifically aerospace engineering. For many times I had regrets and I didn't know what I was going to do. I was like, I didn't want to work with planes and all that. But doing this project, I realized that I can get into planetary science and astronomy. It's actually something that I really got into, and it actually got me into research because this is something I really enjoy doing, and I actually enjoyed the collaborative space I had with my lab, and it's something I plan on continuing in the future and hopefully getting deeper into next summer.
Awesome. Thank you.
Thank you.
All right. Awesome job. Let's round of applause for Anthony. Awesome, awesome, awesome. All right. We're going to keep on moving forward with Naana Asamoah.
Hello.
Hello. Go ahead and share.
Can you see my screen?
Yep, we're there. Awesome. You're good to go.
Okay. Hello, everyone. My name is Naana Asamoah, and I am a rising junior attending the University of Maryland, majoring in biological sciences with a specialization in cell biology and genetics. As a part of the McNair Scholars Program this summer, in the Dye Lab in the Department of Biochemistry, I worked on visualizing AP1 cleavage of A basic sites in the DNA by NMR spectroscopy. So a little background, A basic sites are lesions in the DNA where there is a missing nucleobase. And A basic sites are relevant to us today as they can contribute to cell death. And an example of this cell death is the poison ricin, which is a poison that creates A basic sites in the ribosomal RNA. And additionally, A basic sites can contribute to carcinogenesis, neurodegenerative diseases, and aging. So as known, the DNA is structured in a way where there is a phosphate group attached to a deoxyribose sugar, which is then attached to a nucleobase. When an A basic site forms, this nucleobase is missing, and in its place is an OH group or a hydroxyl group. Now, A basic sites can be cleaved by AP endonuclease 1, or as I will refer to throughout this presentation as AP1. And AP1 is involved in the base excision repair pathway. As you can see here, there is a wrong pairing of the complementary bases where a guanine is paired to a uracil, and in this base excision repair pathway, uracil DNA glycosylase comes in to remove this uracil, and there's an A basic site in its place. AP endonuclease 1, AP1, comes in to cleave this site and leaves a hydroxyl group and a deoxyribose phosphate sugar. So for my research question this summer, I asked if we can visualize cleavage of A basic sites by AP1 using NMR spectroscopy. I hypothesized that since we know that AP1 cleaves A basic sites and leaves a 3' OH and a 5' deoxyribose phosphate, and it makes a change in the chemical environment, this chemical environment change could be amenable to NMR probing at the atomic resolution. So what exactly is NMR spectroscopy? NMR stands for nuclear magnetic resonance, and it is a powerful tool which is used for studying the electronic environment of atomic nuclei structure and interactions of biomolecules. There are many advantages to using NMR, such as the one that I'll be taking advantage of today, which is its ability to visualize the interactions between biomolecules at the atomic level in real time. So as you can see here in the images on the right, there is an ethanol, and we can see the different groups of this ethanol on the NMR spectra. So for my experimental method, I started by forming the duplexes and obtaining a 1D NMR spectra from them. As AP1 has been found to cleave A basic sites, it was important for me to repeat these procedures of past studies so that I can confirm experimental conditions. Next, I added AP1 to the duplexes and obtained a NMR spectrum from them. And I used the NMR spectra I obtained from the duplexes with an A basic site and without an A basic site and compared the NMR spectras. Lastly, I confirmed AP1 cleavage of the duplexes by obtaining a denaturing page gel of the samples, which I will talk about later. So first, I saw that in the DNA duplexes without the A basic site, there was no change in the NMR spectras, therefore there was no chemical shifts. As you can see here in these two images, an overlay of the NMR spectras shows that there's no change in the peak formations of the amino region, which can be seen in the peaks between the 10 to 15 ppm mark. However, when looking at the DNA duplexes with the A basic sites, I saw that there was a change in the peaks of the amino region. Here, DNA-F, DNA-C stands for the DNA duplex with an A basic site, where the F stands for tetrahydrofuran, which is a marker for the A basic site. As you can see here in the zoomed in image of the red box from the left image, you can see that there is a difference in how the peaks are formed based on the time points of when I added AP1 and also with the duplex without the AP1. Additionally, I also obtained a C13 edited NMR spectra of the DNA duplex with the A basic site, since I saw that there was a chemical shift of the duplex in the 1D NMR spectra. As you can see here in the ribose region, in the box, there is a change in the peaks, and when you zoom into that image, as you can see on the right image, you can see that there's a difference in how the peaks are formed, and this basically shows that there are chemical shifts. Lastly, I also confirmed cleavage of the A basic sites by AP1 using gel electrophoresis. Here you can see that in lanes 4, 5, 7, and 9, there are bands at the bottom of the gels, which indicate AP1 cleavage due to the appearance of smaller product bands. These bands are at the bottom of the gel, and this shows basically that AP1 was able to cleave these duplexes. So in conclusion, I found that these NMR experiments we were able to see a chemical shift perturbations in the amino region of the 1D NMR spectra, and also in the ribose region of the 13 edited NMR spectra upon AP1 cleavage. And this was basically an indication of that we're able to see this AP1 cleavage based on the chemical shift perturbations. Additionally, this evidence was corroborated by the gel electrophoresis that I did, where I was able to see the small product bands at the bottom of the lanes of the NMR duplexes with an A basic site, and also with the addition of AP1. So in the future, I would like to use this assay to investigate the effect of AP1 cleavage of A basic sites in the RNA. As known, A basic sites are not really studied in RNA, as there is no base excision repair equivalent in the RNA as there is in the DNA. And it would be very interesting to see how AP1 acts on A basic sites in the RNA. Additionally, I would like to determine the specific atoms within the duplex that are being perturbed by AP1 cleavage using isotopic labeling of the cleavage site. Here are my references, and I would like to thank Dr. Dai and Fran Stump, who are part of the Dai lab, for guiding me throughout this research. And I would also like to thank those at McNair, at UMD, for also helping me throughout the summer with this research. Thank you, and I will now be taking questions.
Awesome job, awesome job, awesome job. Questions? For the portion that you had as your future, the future works with this, are there plans to continue that research?
Yeah, so one of the things that limited my research was the timing and the time constraints that we had. I didn't really have a lot of time during the summer to expand this research, and all those future directions that I had, I plan on continuing this year, throughout this school year, on expanding by getting NMR spectra of just the AP1, which is the enzyme. I would like to get an NMR spectra of that, and also continue with the isotopic labeling with the help of the grad student in my lab.
And so how does this also connect to your graduate school interest?
Yeah, coming into this lab, I didn't really know much about biochemistry research, but I did learn a lot in my chemistry classes, especially about NMR, which we didn't get to touch on in depth in my classes. So being able to participate in this research where I get to use this machine and actually learn how to use how to relate this back to my major, which is more cell biology and genetics. AP1 and A basic sites are along that line where it kind of blends in the chemistry side and also the genetic side. So it was really interesting.
Awesome. Thank you. Any other questions?
I do have a quick question. It's a follow-up to your question, Dr. Hunt, about graduate school interest. How has this research impacted your future career plans?
Yeah, I would say it has probably motivated me to look more into grad school when it comes to biochemistry. I didn't think I wanted to head into that direction in grad school, but coming into this lab and participating in this research, I have found a love of exploring this research in this lab, and I think that it has maybe added a different, I guess, interest that I could look into when looking at different grad schools and what I really want to research when I go to grad school.
Bravo, bravo. This is great. Awesome. Well, round of applause to you and to our all presenters. We thank you for sharing. We are about 5 minutes ahead of schedule. So what we're going to do, we're going to keep going in the same schedule, but moving 5 minutes ahead. So we're going to come back. We're going to take a 5-minute break. So restroom, whatever you need to do. And then we're going to be back on at 9:45 to hear from to begin to hear from our poster presenters. And I believe Isabella is our first to go. So that'll give me a chance to get set to get her going too. All right. So we'll see y'all back at 9:45. Isabella?
Yes.
All right, we're ready to go. Let me share screen. Welcome back, everyone, as we continue the second McNair Symposium here, as our scholars share with the National Science Foundation, as well as the Department of Education, this amazing research that they are doing. I will be sharing screen here. We had some, I don't know, could y'all hear the music? I put some happy on because I wanted us to be happy. And so here we go. Okay. View full screen.
Okay, hi everyone. My name is Isabella Dressler. I'm from the University of Maryland, Baltimore County. And so my project is the exploration of CDAN1's role on RNF168 overexpressed cancers. And so this project was done in the Christ Lab in the Washington University and St. Louis School of Medicine. So most breast and ovarian cancers have extra RNF168 copies. So what exactly does this mean? Well, typically, in terms of copies of a gene, there's typically only 2. However, if you look at the first graph in the introduction section, you can see that according to the Dept cancer database of RNF168 copies, there's typically 3 or 4 copies. And so this means that RNF168 expression is amplified because extra copies can enhance gene function or increase
Isabella, hold on. Michael, you are not sharing her poster right now.
Oh.
There you are.
It says that I am.
You were not before, but now you are. You are good now.
I don't know. I ain't do nothing. So I don't know what's going on. So can y'all see it?
I was able to see it.
We can see it now in the live audience.
Okay, cool. Okay.
Was everyone able to see it or no?
They just continue. I think they were, but yeah, just continue, or you can go back, whatever you want to do. Go ahead.
Okay. So on the graph, you can see typically there's 3 or 4 copies rather than the typical 2 copies. And so extra copies can enhance gene function or increase genomic instability. And so RNF168 is an E3 ubiquitin ligase involved in BRCA1 recruitment through homologous repair. And I'm going to break that down a little bit for you. So RNF168 is an E3 ubiquitin ligase. And so what an E3 ubiquitin ligase does is it ubiquitinates the lysine tail, and so it contributes to the recruitment of repair proteins such as BRCA1. And so with BRCA1, it is a repair protein that is recruited for repair of specifically double-stranded breaks. And so double-stranded breaks is a very toxic form of DNA damage. And so with RNF168, with the function, so it contributes to DNA damage signaling of BRCA1. And so when there is an amplification, it could possibly interrupt this process of DNA damage signaling. It could possibly signal for something else, some other repair protein, and just impact that process. However, it could actually even better do its role of repair. And so this is an issue when it comes to chemotherapy and radiation. And so when it is able to better repair the cells for chemotherapy and radiation, this actually means that the cancer cells can better survive. And so when there's a better survival of these cancers, that is an issue with these amplifications. And so how can we further target this cancer so that we can get rid of all of the cells and kill all the cells? And how do we counteract this issue? So in the Christ Lab, we tried to find a gene that we could further target this cancer to better kill it off and reduce its survival. And so we looked at the online cancer database, DepMap. And so we compared screens from RNF168 amplified cancer cell lines against RNF168 cancer cell lines that were only diploid. The ones on the right, or the genes on the right, have a positive effect on growth, and the ones on the left have a negative effect on growth. Above the blue line of the y-axis is statistically significant genes, and the data in the quadrants were the ones that we felt had a biologically meaningful significance, as well as a statistically significant difference. We chose CDAN1 because of its potential role in the DNA damage response. And so also another reason that we are also interested in CDAN1 is because when we did our own experiment, CDAN1 was one of the top hits. So if you go up to the graph where it has the place of knockout, wild type, and overexpression, we did a whole genome sgRNA library. And so essentially we were taking all of the genes, and so we were knocking them down, and we were assessing their effects on the survival of those genes. And so we also tested CDAN1 just to see what would happen. And so when DNA double-stranded breaks are induced through radiation and hydroxyurea, this shows that CDAN1, when RNF168 amplified cells are knocked down, cells in an RNF168 overexpression background are able to proliferate and better survive and repair from the double-stranded break. And so another thing is we looked through the literature, and so we found that CDAN1 has a connection with the histone chaperone AFS1A. And so it contributes to the DNA damage response through bringing histones to the chromatin. And so chromatin is very dynamic. It goes through structure change during DNA damage response. So aiding in the opening of heterochromatin contributes to recruitment of assembly and repair proteins, similarly to BRCA1. And so we tried to look at CDAN1's relationship and AFS1A's relationship. And so we just found that it is a negative regulator. And so basically for the methodology, we wanted to make a knockout and an overexpression vector. And so we did this by targeting 3 exons for the knockout vector. And so we did PCR amplification reactions for the overexpression vector. And so through there, we wanted to stabilize it by inserting it into a lentiviral vector, into various RNF168 backgrounds, and then analyze it through growth curves and drug testing. So for my results, we tried to see if there's any direct relationship between RNF168 and CDAN1. And so based on the graph, as you can see with the tubulin expression and the not very varying CDAN1 levels, we can see that there's not any particular relationship between RNF168 and CDAN1. And so we tried to see if this was a trend between ovarian and breast tissue cell lines with a gain of RNF168 and just normal. And so we did not see any particular trends identified with normal tissue versus cancer tissue. And so then we chose to start with the knockout vector of exons 2, exons 13, and exons 6. And so on the third Western blot, you can see that 2, 6, and 13, there is no CDAN1 band. And so this is good because the KD is 134. And so no band shows that it was effectively knocked down. So CDAN1 knockout vector, we were able to successfully transduce to lentivirus. And so we had a successful ligation of CDAN1 PCR amplification product. And so we will next ligate it into a plasmid, into a lentivirus. And so we need to do further testing of the growth curves and analysis and drug testing of CDAN1 lentiviral products in order to see if it'll be a potential therapeutic target for RNF168 over amplified cancers.
Awesome. Okay, this is what we're going to, this is what we're going to do. I'm gonna pause for a minute to see if there are any questions, and then we're going to try to do that instead of waiting till all of you go, because there might be specific questions that folks might have. So we have time for one question if folks have a question for Isabella. And with that said, I'm gonna reshare the screen so folks can see. So is there any question? And I can't, because I'm sharing the screen, I can't really see hands or see people right now. So if somebody else come off of mute, that'll be great.
Hi, Isabella, can you talk a little bit about what's entailed in further testing?
So I actually, unfortunately, was not able to get to that with further testing, but by further testing, so we were able to make one of the plasmids for the knockout vector. We were not able to successfully make the plasmid for the overexpression vector, but for further testing, we would insert it into a lentiviral vector for stability, and then the RNF168 amplified cells with those background and a wild type background. And so from there, we would look at the rate in which the cells would proliferate. And so we would make a growth curve. And just depending on those growth curves, we might also look at it for further drug testing to see if it is a potential therapeutic target for these types of cancers.
Awesome. Awesome job. Thank you, Isabella. Round of applause, round of applause. All right. Give me a second because I'm now got to go back to my other screen here. All right, Jaden Martin, you can go ahead and share your screen.
Yes, sir. Yes, sir.
Amen.
Oh, I'm having. Oh, okay, there you go.
There you go.
All right, there we go. Can you see it?
Yes, you're good to go.
Okay. Good morning, everyone. My name is Jaden Martin. I'm a junior mechanical engineering major at Southern University Agriculture and Mechanical College. Today, I'll be giving you an introduction to machine learning assisted polymer property prediction. This summer, I was fortunate enough to participate in a research experience for undergraduates, and I had the opportunity to learn about the world of polymers. You might ask yourself, what are polymers? I'm here to inform you that we use polymers in our everyday life. Things like plastic bottles, things like rubber tires, and sometimes in clothing, you may notice a material called polyester. In household appliances, you may have heard of a PVC pipe, which is an abbreviation for polyvinyl chloride. So the purpose of this work is to increase access to innovative materials while also bridging barriers in polymer design and optimization. So what we're going to do today is, by leveraging machine learning, we're going to aim to revolutionize the way that we approach the development of polymers. What is machine learning? Machine learning, for the average user, I apologize. Machine learning for the average user is a subset of artificial intelligence. Machine learning allows computers to analyze data, to learn from data, and to improve upon the data going forward. Essentially, it uses the algorithms. I apologize. So one of the key advantages of machine learning is speed. Machine learning can do thousands of mathematical applications in a matter of seconds to minutes in comparison to a matter of days to weeks with humans. So let's just imagine that I have to do 500 mathematical applications. It'd be quicker for me to employ machine learning versus I have humans do 500 applications and then I have to go back and verify for accuracy. So the motivation behind this research stems from the demand for polymers that are sustainable, efficient, and high performing in various industries. Traditionally, the design can be time consuming and expensive, which can be, I apologize. Traditionally, the process can be time consuming and expensive due to the experimental nature of testing. So this eliminates a lot of time, so we're saving time, and we're also expediting the delivery process. So before we get started, I want to review the basic structure of polymers. Here in our middle row, we have a monomer. A monomer is defined as a small molecule. A polymer is a combination of monomers. Monomers form together to make a polymeric chain. The arrangement of these monomers affect the physical property of the polymers. Is to predict how different combinations of monomers can lead to different polymers. For example, if I want a polymer that is three things, I want it to be strong, I want it to be lightweight and thermally conductive. Machine learning can help me identify the right combinations of monomers to create the polymer with those exact qualifications. So to prepare to use the machine learning, we employ the SMILES notation. SMILES is an abbreviation. SMILES stands for Simplified Molecular Input Line Entry System. This notation represents the chemical structures of polymers in the format that machine learning algorithms can process. SMILES notation also simplifies the complex structure into a sequence of characters that can be fed into our machine learning models. So next, as we transition to implementing the machine learning models, we exercise our Morgan fingerprinting. The process of Morgan fingerprinting is a technique of machine learning that converts the SMILES notation into a binary notation of zeros and ones. This binary notation of zeros and ones is called a data set. The data set represents the important features of the molecule, such as the atomic connections and the structural details. And keep in mind that the structural details will affect the polymer that is predicted. So in our approach, we use artificial neural networks. The reason why we use artificial neural networks is because artificial neural networks mimic the way that the human brain processes information. Neural networks help us to identify the patterns between the polymer structures and their properties that might not be immediately obvious through traditional methods. So by training our machine learning models on the existing data, we can predict the properties of new untested polymers. I want to highlight a few real-world applications for these polymers. In the medical field, biodegradable stents, they're made to dissolve over time. This can help by causing fewer follow-up surgeries by the patients. And also machine learning can help us to predict the mechanical properties like the strength and the flexibility of the biodegradable stents. And also what's very interesting to me was we were able to predict how fast it would dissolve. That way we can tailor each stent specifically to each patient's recovery process. Polymers are also very relevant in the clean, renewable energy industry. Take wind turbine blades, for example. Machine learning helps us to identify what combination of monomers will produce a polymer that will help us to be able to optimize the strength and the flexibility of the blade in the wind turbine. This ensures that they will be able to endure tough environmental conditions while also maximizing their performance and lifespan. As we result,
1 minute.
Yes, sir. As we result, our results show that machine learning models, when combined with the physical and chemical principles, have the ability to increase accuracy and efficiency. So now that we're able to, not only are we able to predict polymers faster, but we also able to allow for the customization of new polymers. So we're able to create new polymers faster, and we're also able to customize polymers that already exist to improve them. So let's say if I want to improve the durability of a polymer, or I want to make a polymer more environmentally stable, I can use machine learning to improve an already existing polymer. So in conclusion, this research demonstrates how machine learning can revolutionize polymer design by doing the two things that I stated before, reducing costs and expediting the development process. The synergy or the connection between machine learning and material science, it highlights the importance of interdisciplinary collaboration between material scientists, data scientists, and engineers. Looking ahead, we aim to further refine the machine learning models to continue to improve accuracy. We also plan to incorporate a more diverse training data set so that we can have our models able to handle a wider range of polymer properties. And lastly, we hope to develop a user-friendly interface so the industry partners will be able to easily input their material specifications and get predictions in real time. To end, I have a few acknowledgements. I would like to thank Dr. Asamoah and Dr. Yan. I would like to thank the Crest Center. I would like to thank the entire McNair and Trio family, and I also would like to thank the National Science Foundation. Thank you guys for your time. Do you have any questions?
Awesome job. Awesome job. Go ahead and put back up your poster. There. All right. Any questions?
I think Ms. Norman has her hand.
And come off of mute if you do. That's fine.
Question. With these polymers, the human input have to put this into the system correctly in order for the system to be able to manifest and continue to grow. Is that yes or no? So if I was to input the information into the system incorrectly, can the system correct itself and know how to properly input the information, or do a human have to go into it and correct the information so it can give you the correct data that you're actually looking for?
Okay, that's a great question. So actually, initially in the process, when you're training your model, if you implement incorrect information, it will give you an incorrect reading. But on the contrary, if on the initial implementation is correct, the machine learning will notice that, okay, he corrected this. So that's the aspect of learning from the data. So let's say when I initially do it, it's correct. And in the process, I make a mistake, and then I go back and correct it. The machine learning algorithm will notice that I went in and I made that adjustment. So it's going to recognize, okay, that's a mistake. It's not supposed to be like that. So when you're initially training your model, you want to make sure you have 100% pinpoint accuracy, because if you don't, it's going to lead to your later results being inaccurate. But if it's accurate 100% in the implementation and the training of the model, if you make a mistake along the way and you go ahead and fix it, the model will learn that it was a mistake and it needs to be fixed in the future. And that's one of the great advantages of it.
Awesome. Awesome job, Jaden. Thank you so much. You definitely knowledgeable. This was just great. Thank you for sharing. All right, we're going to keep on moving.
We have a question in the room for the live audience.
Oh, okay. Go right ahead.
All right, yeah, you can go.
Okay, so my question is, can you use the same algorithm to test a composite polymer, or is it only specific to a pure polymer?
You can use the same algorithm for composite polymers also. Yes, you can. Awesome. That's also one of the unique aspects of this work.
Okay, my follow-up question is, how would that change if you use a composite polymer, or for the difference that you have with the pure polymer?
Yeah, so the only thing that would really change would just be the training. I would just have to train a different model specifically for composite polymers. So this kind of like problem specific, like with the biodegradable stents. If I have someone with pre-existing conditions, I would have to train a model for that to match with the time, because everybody's recovery process is different, because everybody has a different, it's case by case. So I would have to create a separate model just for the composites.
Okay.
Okay, no problem.
Awesome. Thank you. Thank you again, Jaden. Akriti.
Yeah, I'll be able to share it. Are you guys able to see my presentation poster?
Yes, we can, but are you able to put it in presenter mode where we can see it is small?
Okay.
No, go out of that. Play from start. See if there we go. Okay, there you go.
Okay. Give me a second. Is it still sharing? Because I cannot see my screen.
Say it again. We can't see it. It's very small. If you put it back on the play from start, then we can see it.
I'll try to screen share. That can. screen share.
Polymer condition. So if I want,
Jaden, put yourself on mute there.
I apologize.
Are you guys able to see my screen right now?
Can you hit the play from start at the top?
Okay.
Now we can.
Okay, awesome. So hi everyone, my name is Akriti Mishra. I am a junior mechanical engineering student at University of Pittsburgh, and this summer my research was about analyzing the effects of entanglement on hydrogel osmotic pressure. A little bit of background on each term. A hydrogel is a 3D network of hydrophilic polymers that can swell in water and has the ability to hold a large amount of water while still maintaining the structure due to chemical and physical cross-linking of individual polymer chains, which means that hydrogel also have pressure, which is a driving force of the swelling of the hydrogel that kind of stops the hydrogel from swelling itself, causing that pressure and creating two parts of osmotic pressure. First is mixing part of osmotic pressure, and the second is elastic part of osmotic pressure. My part in the research was more towards learning about elastic part of osmotic pressure because before I joined the research, my professor, Dr. Leo, was already working on the mixing part of osmotic pressure, and we were able to successfully measure the mixing part, and then we were trying to individually measure the elastic part without the help of mixing part. And so to do that, we were able to set up a fully constrained condition leading to a pressure relaxation curve, which is, you can see in the results section, that is like the curve that kind of measures the force, and then we were able to calculate osmotic pressure through it, and then we were able to use that total osmotic pressure, and then subtract it from the mixing part, which was already found, and to find the elastic part of the osmotic pressure. So before I even talked all about it, what is elastic part of osmotic pressure, right? So it is basically in the figure, as you can see, the bottom down, the smaller one, it is caused by the polymer stretching, and polymer stretches when it is cross-linked together. And so on my part of the research was that how do we know that the elastic part is how do we know that the elastic part is really how do we like calculate elastic part like physically quantitatively, like with numbers? Because it was beforehand, it was really difficult. It was hard to be studied, and we did not know much about how do we there was not a lot of understanding of elastic part of osmotic pressure itself. So some of the challenges we dealt with was that elastic part beforehand on some of the old established model like Fleury-Renner model, it did not fit with the Fleury-Renner's model, which also was working on calculating mixing part of osmotic pressure, but they did not encounter the elastic part, which made it more difficult for us in a research group to know how does in a polymer, how do we know that it's consistent throughout every level of water content. So like in a gel, we can add certain water content. So in the higher level of water content, the mixing part was correct even with the elastic part of osmotic pressure, but in a very like small soft gels, we don't know if it's really as effective, and how does the elastic part affects the elasticity of hydrogel itself, which can be used in a lot of areas and applications like hydraulic fracturing and water sort-off operation, which is also related to my area of education, but also it's very useful in biomedical field like medical dressing, monitoring drug deliveries, wound dressing, and so on. So in the hypothesis, when we set up the fully constrained condition, some of the things we hypothesized was that the degree of entanglements is actually related to the initial polymer content, and initial polymer content is the initial amount of monomers and cross-linking the chemicals that we add, and then the final polymer content, which we just call polymer content, is the final weight after everything added and then dehydrated from a certain percentage of water to another percentage of water. So we hypothesized that really it might be the level of entanglement that happens, as you can see in the diagram. I don't know if I can scroll down, but the diagram that has like a cylindrical shape, it can show that there's different links to it. And then when networks, the polymer chain networks are connected, they might get like entangled, and that might be one of the cause of change in elasticity. And we did not know about it as before, and we were not able to, that's why we were not able to analyze tiny, smaller levels of gel. So another hypothesis was that because measuring elastic part of osmotic pressure, pi Ella is like the annotation in the poster I have, was such a big challenge. There might be that we can control the entanglement behaviors in hydrogel. So and to do that, we were able to measure different levels of initial polymer content, because they're related to the initial, because they're related to the entanglement. So we measure the initial, we use different initial polymer content, and we also were able to use compare different various final polymer content with measure of the elastic part of osmotic pressure. As you can see in the final result, I have a bar chart diagram and a normal graph, where the first diagram talks like kind of shows that with
1 minute.
yeah, okay, with like lower level of, so the less water it has, which means the more polymer content it has, the higher elastic part of osmotic pressure it is. And on the other side, the graph shows that even though there might be true for one sample, but what if there are different level of final polymer content? So in that graph, you can see that there's three initial polymer content with a different level of final polymer content, and they all kind of like are synced together. Towards the end, it might like we were struggling to study the results, but there might be technical issues that we were trying to fix later on. And my PhD student who I was working under was able to like analyze, and he's still trying to understand and study the further reason of why the graph shows a little bit slight change. But overall, the observation was that a higher degree of entanglement leads to higher elastic part of osmotic pressure, and then the tendency can also be observed at the various final osmotic final polymer content, and then it kind of like the elastic part of osmotic pressure kind of increases with the initial polymer content. And so future work, we are hoping to measure the elastic osmotic pressure with the prediction of the Fleury-Reynolds model to like kind of update the information. And then we are hoping that we will know more parameters like the phi, that is final polymer content, and initial polymer content, and with different cross-linking densities. So yeah, that was the research that I was able to work. And I would like to thank my mentor, Dr. Leo, and Zayfan. He is a PhD student. He was able to help me throughout the journey of the research. And I would like to acknowledge the McNair Scholars Program for sponsoring and helping me throughout this journey.
Awesome. We got time for maybe one question. Someone have a question? This is great. Wow. Well, I'll ask you a similar question I asked a student earlier is how has this research sort of impacted your desire for graduate studies? Is this something you're interested in doing further work on yourself?
So this is my second research of my whole, from what I have done throughout my bachelor's degree. I wanted to do this research specifically because I wanted to get experience in laboratory. So we were able to just go and do hands-on work and make gel. And I like that part of research, but moving forward, I would like to get more research experience on aerospace engineering, as one of the other presenter, Anthony, was able to present. So yeah, that was something I was able to get a lot of experience, and I enjoyed this research. But considering that I do like to do hands-on work now, I would like to kind of save the same experience but work towards my major.
Awesome, awesome. Wonderful. Well, thank you for sharing. I didn't, was there any other question or one before? Was there another question? Making sure I didn't miss anybody. All right. Well, congratulations. Thank you again for sharing. Round of applause. Wonderful job. Wonderful job. All right. We're going to keep on moving with Nolan. All right.
Can you all see that all right?
Yep. Good to go.
Great. Thank you. Hello, everyone. My name is Nolan Gerdes, and I'm a senior at the University of Wyoming, majoring in civil engineering. Today, I'll be presenting my research entitled Evaluating the Recovery of Hydraulic Conductivity in Fire-Affected Soils. Wildfire research as a whole has become more important during this age because of climate change. Climate change is increasing the frequency, extent, and amount of wildfires happening all over the world, especially here in the United States in the Rocky Mountain region. And these wildfires have ramifications beyond just what happens during the wildfire. Following wildfire, plant cover is burned, soil properties are changed, and soils show water repellency. And as these wildfires happen, these areas that are affected are vulnerable to extreme flooding and erosion events, which can impact downstream water quality, be a hazard to human lives, and destroy infrastructure. So the hydraulic recovery of these soils that have been affected by wildfires is an important topic now in post-wildfire hydrology research, because it looks at how we can restore an ecosystem's ability to protect the human lives and come back to its pre-burn state. So the focus of my research was to look at infiltration following wildfires. And there had been previous studies that had done this. Some had found that infiltration increases with time since burning, and there should be a return to pre-burn conditions within 6 years of burning. However, these studies were limited because they compared soil from different areas of land. So they sampled burned soil and then compared that to unburned soil in a different area, which makes it difficult to draw comparisons. Additionally, studies that did compare burned and unburned soil from the same location were only limited to 1 year. So there's a serious gap in knowledge. And how I address this is I used slash and burn piles, which are a series of piles burned over winters, as you can see on the picture on the left. And these are a proxy for wildfires, as they are burned at the same level that a wildfire is. And to investigate infiltration, I looked at the hydraulic conductivity of these slash and burn piles. Hydraulic conductivity is a component of infiltration and controls the gravity component of water moving through a soil. So with these soil samples, I started with kind of reverse order here, pile 10. I wanted to see if within these piles, there was variability in the hydraulic conductivity. And what I found was there wasn't variability. So in the following 9 piles, I was able to just take 3 sample points within the piles and then 3 sample points outside of the piles. And this allowed me to have paired unburned and burned measurements. I collected a total of 12 inside that first pile, and then a total of 3 inside the other 9 piles. And I compared this data to one another using the Wilcoxon rank sum test, which determines statistical significance between the means. What I found following this statistical analysis is that hydraulic conductivity was not significantly affected 1 year following wildfire, which was different from previous research, which showed significant decrease in infiltration and hydraulic conductivity. There was, however, a significant change in hydraulic conductivity 3 years following wildfire. So instead of seeing an increase in hydraulic conductivity over time, we observed a decrease and further deterioration of hydraulic conductivity following burning. And this suggests that there's an interim process between years 1 and 3 in which some sort of soil sealing happens. I wasn't able to explore this further because of the time restraints on the field research component of my project, but it is something I'm looking into in the future. Additionally, only the topmost layer of these soils was affected by the burning, and this aligns with a study by Ebel and Moody in 2017, who also witnessed the same amount of burning in a wildfire. So this showed that our slash and burn piles served as a good proxy for wildfires. For future research, it would be interesting to look into sorptivity, which is another component of infiltration, and the ratio of sorptivity squared over hydraulic conductivity. This had also been investigated by Ebel and Moody, and they found that the relationship between sorptivity and hydraulic conductivity was a better predictor of a landscape's vulnerability to these overland flows and debris flows. So overall, I found that slash and burn piles significantly alter the of soil following burning. I'd like to thank the McNair Scholars Program at the University of Wyoming, the Department of Civil Architectural Engineering and Construction Management from the University of Wyoming, the NSF, which my PI was funded by, my PI, Dr. Ryan Webb, Corey Mooney, Nana Frimpong, and Dr. Glenn Liston. Thank you all for listening. Does anyone have any questions?
Awesome job. Questions?
I had a question. Amazing job. I had a question on, do you have any theories on what happens during that second year and the soil sealing process?
Sure. So 1 year post-wildfire, we observed that there was a significant layer of ash on top of the soil that hadn't condensed into like a single layer yet, which allowed water to flow through it. Whereas the piles burned 3 years previous, that layer was gone and it was deposited within the soil pores of the actual soil. And I think that all of that ash got into those soil pores, and because of ash's inability to absorb water and the lack of space in those soil pores, it didn't allow water to infiltrate into the soil.
Have you thought about going deeper, doing like a deeper soil core?
By deeper, do you mean like deeper into the soil or like?
Yes,
Okay.
doing like a core and trying to look at like an entire profile of the soil.
Sure. Yeah, we dug a pit in each of the piles to look if that was necessary. And it was clear in all the piles, only the top 2 centimeters of each pile was affected by the burning. So a full soil profile wasn't necessary for our experiment.
Awesome. Awesome. Thank you, Nolan. Round of applause for Nolan.
Thank you all.
Thank you. Thank you. And now we will have, I think we have one more person. Kayvona.
Yes.
And I think you should be able to share your screen. Go for it. There you go.
All right. Hello, everyone. My name is Kayvona Brown. I'm a senior at the University of Connecticut studying psychological sciences with minors in sociology and women and gender sexuality studies. My project today is about the relation between PTSD symptom clusters and pain within a recent sexual assault survivors with an examination of racial differences. As a sexual assault survivor myself, I find this very important to advocate about and also just to spread my advocacy in my future research as an aspiring clinical psychologist. I do want to present a trigger warning. This topic will be about sexual assault and rape, and I know this is very sensitive. So please do engage in self-care. After experiencing sexual assault, a survivor may experience post-traumatic stress disorder, also known as PTSD. Post-traumatic stress disorder is a disorder that causes unwanted behaviors, feelings, and physiological hyperarousal, which is basically when your body's in a state of fight or flight mode due to the trauma that you've experienced. Also, PTSD is organized into four symptom clusters: PTSD cluster B, which is re-experiencing the trauma; PTSD cluster C, which is avoidance of the trauma; PTSD cluster D, which is negative altercation in cognition and mood; and PTSD cluster E, which is increased arousal and reactivity. I bring this up because PTSD is often researched within the field at the syndrome level, which means that all these these clusters are analyzed together, rather than analyzing these clusters individually by the symptom clusters that affect the patients that are having these things. I bring this up also because PTSD symptom clusters is very important to analyze when it comes to reviewing or looking over like how it affects health behaviors such as, or health effects such as pain. PTSD and pain have been linked and have been shown to be very highly comorbid, in meaning that they both affect each other. And it's a bi-directional relationship. This is important because it's important to analyze the symptom clusters because within research, they only analyze the PTSD cluster, the PTSD syndromes and the pain within veteran populations and within male populations, and it leaves out other populations that need to be examined, such as women. Within my project, I analyze PTSD symptom clusters separately, and it's linked to pain with an examination of racial differences within women population. I bring up racial differences because it's understudied within research as well. I use the theoretical framework of the fear avoidance model, which I'll sum up for you guys, is that PTSD symptoms, such as all the four clusters that I have spoken about, can affect the way that the pain symptoms show up. So they exaggerate it. And it's a bidirectional model, which also says that the pain symptoms that an individual may feel can also exacerbate the PTSD symptoms. So for my hypothesis, I have three. One states that overall, the PTSD symptoms and pain will be positively associated against all women of sexual assault survivors. PTSD cluster D, which specifically says that hypothesis 2, which specifically says that PTSD cluster E, which is increased arousal and reactivity, will have a positive correlation with pain and other system clusters. And then hypothesis 3, which states that racial ethnic minorities will have a higher correlation between PTSD and pain than white individuals. For my methods, I draw this study from a larger longitudinal study, which had 203 women sexual assault survivors, and recruited this through an online platform called Prolific. We used self-reporting surveys to assess the PTSD symptoms, the pain severity, which is basically how severe is your pain on a day-to-day basis, and then also pain interference, which is how much does your pain interfere on a day-to-day basis. I pull out also that we took this information 1 month after the trauma has occurred. So this is a very niche thing. It's not really studied in my field often. We often look at the research with years, or we often look at the trauma years after it has occurred. For the results, hypothesis 1 and hypothesis 2, hypothesis 1 and hypothesis 3 was very consistent. However, hypothesis 2, is inconsistent. It's partially correct, but not correct. And I bring that up because PTSD cluster B, C, D, and E were all positively associated with pain interference. So there was an interference within their day-to-day because of their pain, but only PTSD cluster D was associated with pain severity, stating that there was something, that the pain was severe on a day-to-day basis. If we take a look at graph 1, you can see that racial ethnic minorities are in Black, and then white individuals are in beige. And you can see that PTSD overall showed that racial minorities had a higher rate of PTSD symptoms overall compared to the white individuals. And that was the same for most of the other clusters as well. If we take a look at graph 2, you can see that pain interference and pain severity were the 2 measures that I was measuring within the racial groups, with racial ethnic minorities being Black and white individuals being beige. You can see that it's pretty consistent overall. To sum this up, women sexual assault survivors who suffer from PTSD have significantly higher levels of pain severity and pain interference, which was something that was shown already in previous research. And it was also supported by hypothesis 1. But however, for hypothesis 2, it is revealed within literature that PTSD cluster E was positively associated with pain. But within my study, only PTSD cluster D was associated with pain severity. This is very important to point out because of the time frame in which we took this information from. It was 1 month after the trauma. So PTSD cluster D is negative altercations in cognition and mood. So that means that you're experiencing symptoms of like helplessness or negative thinking, and that alone can exacerbate the way that you view your pain in general. And it's a bidirectional model because it goes back to the fear avoidance model that states that if not only will your PTSD symptoms exacerbate your pain, but if you have high pain severity already, it will also exacerbate the way that you view your PTSD symptoms. And overall, when we go back to hypothesis 3, that states that racial minorities will have a stronger association between pain interference and pain severity, that was true. And if we look back into literature, it states the reason why this is the case is because racial ethnic minorities are more prone to being exposed to trauma more than white individuals, which adds factors to their PTSD symptoms, and it causes their pain to be a little bit more exacerbated than white individuals. So I took on the study because I found it very important to not only point out these differences between demographics groups, but just to point out the importance of analyzing within these groups separately as well. Because when we just analyze PTSD overall, we don't necessarily get a deep inspect to those symptoms that each individual is actually occurring or going through and how that affects with other health situations such as pain. I would like to take the moment to say thank you to all of the people that have supported me, Dr. Renee and the McNair program at the University of Connecticut, my PI, which is Dr. Crystal Park, and my 2 project mentors. And I would like to also say thank you to the NSF for allowing me to share my story and to share my study with everyone. It's very important that people get to know a little bit more. I know this is a sensitive topic, but it does affect a lot of women around the world, especially on college campuses. And I think it's very important to advocate this as well. My future goals is to take this information and not only to expand on it, but to look at it through a longitudinal study, because this was a cross-sectional study, so I don't get to actually look at this and state my statings and feel like 100% confident about it. I think I need a little bit more information to kind of push it. And I also would like to write a manuscript on this and to get a little bit more in a deep dive on the racial differences and how PTSD really affects individual groups at that level. And that was my project. Any questions? Oops. Do I have any?
Awesome job. Awesome job. Questions? Wow. I just want to also lift up what someone even has sort of shared their thank you for the bravery to do this work. So definitely I want to lift that in this space. Because this is both important work and needed, especially and I appreciate you as a Black woman doing this, even though I know it's also on the shoulders of Black women. Like, you know, and so I honor you in taking on this. And it's sad that you have to, if that makes sense, right? You know, so definitely thank you. Any other comments or questions?
I had a there's someone in the chat. I think you should get that.
Okay. Oh, thank you. Okay. And then we'll come back to you. How do you think the time in which you collected the data affected your results?
Yes, this is a very important question. Because it was 1 month after the trauma has occurred, it needs to be studied a little bit more, which is why I wanted to look at it through a longitudinal study instead of a cross-sectional, because the short period of time can make the symptoms that each individual has exaggerated, meaning that it could be way more significant. Think about it as if you got your toe stubbed, and in that moment, it really hurts. And maybe like the day after, it's going to hurt, and the day after, it's going to hurt. But in a week from now, the symptoms probably won't be as bad as it was on day one. So that's why I kind of want to look over it as a within the longitude study itself, because there's 3 time periods, and I only got to do data on one. So if I looked over it over the next couple of weeks or months, I think I will see a consistent, but also just a little bit of a difference, maybe in the PTSD clusters itself, but maybe not in the PTSD symptoms overall and its effect to pain severity and pain interference. I feel like pain interference will show and reveal that it's still like very consistent, but pain severity will probably decrease. And that's just based off of my hypothesis, not what research has shown.
Awesome. Thank you. Eve, did you have, I think that was Eve. Did you have a question?
Yeah, but if there's anyone else, because I've already asked questions, I'm happy to.
Oh, no, go ahead. You'll be our last one.
Thank you so much. I mean, incredible. I'm wondering about ethics and data collection and how you feel about that. I mean, this is obviously really important work, but it's also done with really vulnerable people, and how you handled that and how that was emotionally for you and also the participants.
So before the participants can agree to doing the study, we also go over the consent form, which I think most studies do. And it kind of like informs that at any point, if you're uncomfortable, you can leave the study. So, and it's a very daunting task to talk about your experience. So we do create a comfortable space for each participant, and we do allow them to withdraw at any point. So everyone who was in this study was willing to share their story, which I found was very brave, and wanted they wanted people to know what was occurring within their bodies and within their minds. And I thought we just went with it, honestly. Anyone who was uncomfortable had the opportunity to leave, and we didn't use any of their data.
Absolutely, totally makes sense. Like, of course, I just, wanted to know about the system. Thank you so much.
Oh yeah, and the system it's basically that, yeah.
Awesome. Well, thank you so much. Thank you. Round of applause. All right. We're going to take a moment. We're going to take maybe a 5-minute. What time? I think we're sort of on time. We might be a little bit behind, but I think we're doing good. So let's go ahead and take a 5-minute break for you all to get yourselves together, and then we will hear from our NSF leaders. Y'all were phenomenal. Thank you for your attentiveness. Thanks those who are in the hotel ballroom. Thank you for being present. Thank you to all others who have joined us virtually as well. And so we're going to come back on at 10:55, and I'm going to turn it over to our NSF leaders who will share information to you about upcoming opportunities.
That sunshine in my pocket, got that good soul in my feet. I feel that hot blood in my body when it drops. Ooh, I can't take my eyes off it, moving so phenomenally. You're more like the way we rock it, so don't stop. And under the lights, when everything goes, nowhere to hide when I'm getting you close. When we move,
which is the Graduate Research Fellowship Program, and is a funding opportunity from the National Science Foundation, or NSF. And as an overview, it's just going to kind of give you some highlights and then allow you to ask some questions. And if you want more information, you want to explore this opportunity, probably the best place to go is to this website that's right in the bottom corner of the screen in terms of the information. And go to nsfgrfp.org, www.nsfgrfp.org. So welcome from the National Science Foundation, and especially from the Graduate Research Fellowship Program. Ren, could you go to the next slide, please? So here's a quick overview of what we're going to talk about. First, I'm going to just explain a little bit about GRFP in terms of general information and kind of the reason that we have this great option for people to consider in terms of receiving funding for graduate school. And then I'm going to talk a little bit about who's eligible and the eligibility rules, and also the kinds of degree programs and research areas that are not funded, because we don't want you to get all excited but find out that this happens to not be the best option in terms of investing your time for filling out an application for funding. And then we'll mention a little bit of information about the application package, and also about what the reviewers who look at the applications that are submitted to NSF are looking at in terms of evaluating the applications. So with that, let's go to the next slide, and I'll give you a little bit of background about the overall goal of GRFP. So the overall goal is to recruit individuals into science and technology and engineering and math fields. And the idea is to select, recognize, and especially financially support early career individuals with demonstrated potential to be high-achieving scientists and engineers. And an important second goal is to broaden participation of the full spectrum of diverse talents that society has to offer, which includes underrepresented and underserved communities. Those are the main goals. Now, a little bit about GRFP. It's been around since really the start of the National Science Foundation. So the first year that funding was provided for NSF by NSF was 1952. And since then, there have been many, many fellowships, over 70,000. Some people have gone on to get Nobel Awards. There's lots of great stories. The main idea is that it's a way to get $37,000 of stipend for 3 years, and to get a cost of education allowance that's sent to your graduate school. And at that university, if they accept you as a fellow, then they are going to, for those 3 years that you have the stipend, you're going to be able to get a waiver on the tuition and fees. Over each year, we have about 12,000 people that apply, and about 2,000 fellowships are offered. So there are quite a few fellowships that are offered, and you can see the major fields for the in science that are funded. And we've heard about some of those today with some of the talks that we had earlier today. So for example, if you're going into chemistry or computer and information sciences or engineering or geosciences or life sciences, materials research, mathematical sciences, physics and astronomy, psychology, social sciences, and STEM education, this is a potential funding opportunity for you. And it's a funding opportunity if you're going to be enrolled full-time in a research-based master's or doctoral degree. And a lot of people are surprised that this can be a way to fund master's degrees. And so that's really important to remember. Again, there's a lot more information. If you go to nsfgrfp.org, you'll find all sorts of great resources. And so what I'm going to do now is kind of give you a highlight over some of the main things that are good to know about it to begin with. So if we go to the next slide, please. Remember, it's $37,000 for stipend and then $16,000 for education allowance. That means each year, the total amount that NSF sends to your university is $53,000, or the total fellowship amount is up to $159,000. There's also some other opportunities that are available if you're a fellow, including internship opportunities and other kinds of funding that are provided by NSF. So we wanted to just remind you of that there are other things connected with the fellowship along with the $159,000. Next slide, please. So here are some of the kinds of things that are the kind of the features or characteristics of the fellowship. So you get to choose your project or an advisor and graduate program. That's part of the application process or once you get into grad school. And there's no service requirement connected with this. That means after you're through with your graduate education, you don't have to have any service component, for example, working for the government or something like that. There are some fellowships that are like that. And you can use it in any accredited nonprofit U.S. institution of higher education. So those are universities with campuses in the U.S. And as I mentioned before, it's for research-based master's and doctoral degrees. Now, if you're interested in this and you look at some of the other information and listen to what I say later on, you'll know that there's a deadline coming up, for example, for this year for funding for next fall. And you need to check that and connect that, match that with the major fields of study. Your major field of study will have a very specific deadline, and I'll describe that in a little bit of more detail. But it's really important to understand that when you're filling out the application, you're going to choose a particular field that you're going to apply in, and that's the area that you're planning to go to graduate school in, and that's the field that the reviewers who look at your application are experts in. So it's all connected, the deadlines and where you're going to grad school or planning to go to grad school. So those are some of the characteristics. Now, who can apply? You need to be a US citizen or a national or a permanent resident. And of course, it's for early career people that are undergrads that will be going to grad school next fall, or first year or early second year graduate students. Again, you can be working on a master's or a doctoral degree, and that means no professional degrees. And I'll explain that a little bit more detail, just so you get the difference between the research-based degrees and the professional degrees. Hey, it's the National Science Foundation, so it has to be science, technology, engineering, or math. I mentioned some of the fields already, and you have to be enrolled full-time in that graduate program, okay? Now, when you apply, there are different categories for the people that apply, and they have different rules connected with being able to apply. And you can find a lot more information, as I mentioned, on the website. You can also go to our instruction manual, and that's called a solicitation here at NSF. And that number, if you want to check it out on the internet, is NSF 24-591. So I'm giving you kind of a overview of what's provided there. So for example, if you can apply as a senior who's going to be going to graduate school next fall, okay? And as long as you're in that category or a person that has graduated with their bachelor's degree, but has not started graduate school, as a level 1, you can apply to GRFP as many times as you're in that category. So for example, if 2 years from now, you decide to go to graduate school and you still haven't been in graduate school, you'd be a level 1, you could apply, and you could apply again and again as long as you're in that category. Now that's different than if you're in graduate school. You can apply as a first-year graduate student, we call that a level 2, or at the beginning of your second year, and we call that a level 3. If you're a graduate student, if you're in graduate school, you can only apply once. So you might have applied while you were an undergraduate. You can do that as many times as you want. Once you're in graduate school, you have one chance. Now, there's another category that I'll mention, even though probably there aren't a lot of people in the audience right now that this would be connected with. But let's say you were in graduate school for a while, and then you left for at least 2 years, and you didn't have a doctorate degree, and you wanted to come back to school. You're not enrolled in a graduate program. There are conditions under which you could even do that. So you can see there are different chances depending on exactly where you are in your academic career. Again, a lot of details. You can go to 24-591 to learn more about that. Okay, next slide, please. Now, I mentioned that there are some areas that aren't funded through this program. And again, you can go to the solicitation for that. But I mentioned, for example, that we fund research-based master's and doctoral degrees. And that means if you're thinking about going on for an MBA or something like an MD degree or a law degree, a JD degree, those aren't funded through this program. And even if you were going to do a PhD and a JD or a PhD and an MD, those aren't funded through this program. And then there are certain other kinds of degree programs that GRFP doesn't fund. For example, community global public health and counseling and social work and education and humanities, except for education, if it's STEM education, right? Science or math or education, engineering education, that would be okay. And the history of science also is an area where you could get a fellowship. So that's a little bit about degree programs. Now on the next slide, there's something connected with this, and that's usually the degree programs are kind of connected with the research you're planning on doing. So if you're doing research that's directly health related, has directly health related goals, or some types of outcomes research, or work on plant pathology or food safety, those are areas that, again, we're not really funding through this program. There are plenty of areas we are funding, but if you're interested in doing research on treatment of disease or disease prevention or public or community health, or things like health policy, or again, as I mentioned, plant pathology, agricultural production, those kinds of things, you're gonna have to look someplace else for funding for your graduate school, not GRFP. All right, let's go to the next slide. And if after all that you go, well, I'm pretty interested in this, what do I actually have to do? Well, the application is online. And for example, this year, the deadline is coming up. It's coming up in October, and I'll show you a schedule. And these are the different parts of the application. First, when you go online, you'll find that there's a bunch of information you put in about your education, any work or research experience you have, the kinds of honors you might have had, or publications or awards. So kind of some general information like that. And then a lot of your time and effort will be put into 2 statements that you need to write. There's a 3-page personal relevant background and future goals statement, and then a research plan statement. And as I mentioned, this is sort of connected with, if you're not in graduate school already, applying to graduate school, because similar types of statements are often part of the what you need to prepare for going to graduate school, for applying to graduate school. So you can use a lot of the effort you put into the GRFP application also for the kinds of things you need to be thinking about and working on for applying to graduate school. We also need your transcript, and those would be both your undergraduate transcripts and then any graduate school transcripts if you have been in graduate school. And then a really important final component are the letters of reference. And you have an opportunity to list 5 names of people that would potentially send letters to NSF as letters of reference. You have to list 3 names, and at least 2 of those names, those people have to send in letters. And it's really important to be thinking about this. If you're thinking about applying this year and you haven't gotten on to the electronic application system yet, I really urge you to do that, because you need to at least do this part pretty quickly and put those names in there, because NSF's going to send emails notifying those people that you have requested that they send in a letter, and they need to get those letters in by the deadline of October 11th. You have really until the next week after that, but they need to get those letters in pretty quickly. And you obviously want to give them as much time as you can so that they can do, write some really nice letters. Okay, so those are the basic parts. I'll describe a little bit more about some of this, what people are looking at when they look at this package, but that's the basic set of things. Okay, let's go to the next slide. And I mentioned these deadlines. Remember, one thing I said is that depending on what field of study you're going into, you're going to have a slightly different deadline. So you have to be thinking about what graduate program you're going to be interested in. And depending on that field of study, there's a different deadline. For example, this year, if you're going into chemistry or geosciences, and we had some talks about those kinds of fields, the deadline is the 15th of October. There were also some talks that might fit into life sciences, and that would be the 16th of October. And some of the talks might even be part of like psychology or social, behavioral, and economic sciences, and that's the 18th. So you really have to be thinking about matching your field of study, your graduate program, and then these deadlines. And these are deadlines for submitting everything except those letters of reference, which have to be in the week before, right, on the 11th. So it's not difficult to overestimate, emphasize this. You need to check the deadlines, right? You don't want to miss the deadline for your field of study. All right, let's go to the next slide, please. So here's kind of the basic schedule. I mentioned the letters of reference are due on the October 11th. That's not when you need to notify somebody that you want a letter. That's when they need to send it to NSF, okay? And then the week after that, you saw the schedule. We just talked about that. And then if you're not in graduate school yet, if you're planning on going to graduate school, obviously you have to apply, right? So sometime after October, the deadlines start coming up. Usually, well, there are different deadlines at different places. So you need to be doing that. But a lot of the stuff you've been working on with the GRFP might be adapted or used in one way or another for those applications, right? Now, the applications are reviewed, and I'm going to talk a little bit about that. But basically, they're submitted in October, and then we hope that by March or April, we'll be able to notify people of the results, and everybody gets notified. And the people that are offered a fellowship need to let NSF know by May. There's a deadline that will be posted in May. And that's because it'll take us here at NSF some time to get the money over to the university so that you can start using the fellowship money next fall, right? So that's the basic timeline. So if you're thinking about doing this for this year, you need to be getting on this. Maybe some of you are already really busy working on it, and that's good. And if you haven't gotten those letter notifications sent in, boy, you better do that quick. Okay, so that's the timeline. Let's go on to the next slide, please. So now when the reviewers look at the application, they look at everything, right? They look at those statements, the letters, they look at the transcripts, all the information. And NSF, for any proposal that comes to NSF or these applications, we're asking the reviewers to use the National Science Board Merit Review criteria. And there are two things to remember about this. One is they're going to be thinking about it in terms of intellectual merit, and then also broader impact. So when you write those two statements, you better have those headings: intellectual merit, broader impacts. Intellectual merit basically is how important is the proposed research to advancing knowledge? Okay, think about it in that way, advancing knowledge. And broader impacts, that's about the potential benefit of the fellowship or you as an individual for this particular application to benefit society or advance desired societal outcomes. So the reviewers are thinking about this in terms of the demonstrated potential, and they're looking at it in a comprehensive way, looking at everything you send in, and they're asked and instructed to give balanced consideration to all parts of the application. So you can find information and evidence about intellectual merit and broader impacts in any part, potentially, of the application. So it's really important that you send in a very strong application with all parts. Next slide, please. Intellectual merit, what do we mean by that? I mentioned that it's really about this idea of advancing knowledge. What would be some examples of that? Well, it could be the transcripts, right? It could be your grades, or it could be in that first set of information you give in terms of awards and presentations. Or it could be information you provide about your experiences planning and conducting research, or working as a member of a team, or working independently and or communicating research. It could be evidence in your application about you taking the initiative of solving problems, of persisting. Those are the kinds of things we mean by intellectual merit, okay? It also could be specifically about the potential of your approach for advancing knowledge in your field or generally. So that's just a little bit more background about intellectual merit. Now, what about broader impacts? In the next slide, we show you a little bit about that. Broader impacts, remember, that's the potential of the individual, that would be you if you're applying, or the research you're proposing to do to impact society, okay? And these are just some examples, just to kind of give you a feel for what broader impacts are about. For example, when you remember, one of the goals of GRFP is to increase participation of the full spectrum of diverse talent in STEM. So that's a broader impact. Or if you have experience or have plans to engage in mentoring or increase public engagement with science, or conduct outreach of different kinds, or even increase collaboration between like what research is going on in a university and what's going on in a business or industry or in government, or those kinds of things. Those would be broader impacts, right? So think about that kind of stuff. All right, so that's a little bit about intellectual merit and broader impact. If we go to the next slide, please. Just as a final word, if you're working on your application now, you're in good shape. You should always, if you're going to start next year for applying next year, if you're getting ready, start early, okay? And what I mean by that is, look at that solicitation, go online, look at the application module so you get a feel of the different parts of that. Remember that for this year, the letters are due on October 11th. That's not too far away. So you need to let people know you want them to get those letters in. Make sure that the information on your transcripts and the information you provide in all these other parts of the application where they ask for certain kinds of information, make sure they match, okay? And if you're working on both a master's and a bachelor's degree at the same time, sometimes people are in a joint program, then it's really important to think about getting a letter from your registrar at your university confirming that. That kind of information, the details with that are again in the solicitation. And if you happen to be a graduate student, we suggest you send in your official transcripts. There may not be too many people in this group that are in that category, but it's very important that the information match, basically, and that's why we say double check the transcripts, okay? Now, if you need accessibility accommodations to request that, really you should be doing that even before now for this coming cycle for the application deadline in October. If you're doing it next year, remember that about requesting the accessibility accommodations. So that's some background. I hope we have a little time to answer some questions. And it's an amazing opportunity. I hope you'll think about if it fits into what you're thinking about for graduate school.